So our next speaker is Bo Zhang. I hope I said that okay. And Bo obtained his PhD degree in 2019 from South China Sea Institute of Oceanology, the Chinese Academy of Sciences in China. Now he works as a postdoctoral researcher. His research interests in the developmental biology of seahorses. In his study, by comprehensively analyzing the crucial posterior body plan genes, the Wnt gene family, in the lined seahorse Hippocampus erectus, he found a special expression pattern change of Wnt8A during seahorse tail development that may be associated with the elongated tail formation of seahorses. Today, he'll be presenting one of his recently published papers titled Wnt8A is one of the candidate genes that play essential roles in the elongation of the seahorse prehensile tail. So, Bo, are you presenting live or do you have a video you're going to play?
Okay, Bo, well, I will play the video for him.
Excellent.
Okay. Wait a minute.
Hello, everyone. It is my great honor to share my work with you. I'm Zhang Bo. I come from South China Sea Institution of Oceanology. The topic of my presentation is Wnt8A is one of the candidate genes that play essential roles in the elongation of seahorse prehensile tail. My presentation organized in the following 4 parts. Now, let's start with the first part, the background. As you know, seahorse is an interesting marine fish. It belongs to the synthetic hippocampus. Seahorse is well known for its unique characteristics, such as the special blood punch for male pangolins, its body covered with bony plates, and the elongated table snouts, and so on. In this study, we will focus on another interesting trait, the elongating and prehensile tail. Unlike most of the common fish, whose tail is stubby and cylindrical, in seahorse, their tail is an elongated square prism. Their seahorse tail composed of several ring-like segments that ensure it's traceable. The length of the tail account for almost half of the body length in seahorse. Compared with the typical fish, zebrafish, and melaka, the ratio of tail to trunk smith is much higher in seahorse. That indicates a special tail smith genesis may take place during seahorse tail development. Seahorse embryology studies have shown that the special tail morphogenetic features established during embryogenesis. According to previously studied, the development process of seahorse embryo can be divided into 4 sequential stages. The seahorse tail development process occurs throughout the 4 stages. At the early embryogenesis, following their primitive streak formation, the tail primordium is established at the following stages. Many focus on tail bud outgrowth and tail smith genesis. However, the genetic mechanism involved in this special tail development is largely unknown. As is well known, the Wnt gene is extensively studied for its role in body plan. In vertebrates, the mutation or adaptation of Wnt genes often cause several body defects. By determining the fate of the tail progenies and the stem cells, Wnt genes modulate the vertebrate tail morphogenesis. Previously, studies have shown several wind genes involved in vertebrate tail development process. In zebrafish, the wind 8A is a key factor in determining the entering post body formation. The mutation or gene knockout of wind 8A in zebrafish caused the absence of all the segments behind the trunk. Besides, also, other studies have shown that wind 5A and wind 11 also involved in zebrafish tail development. The double mutation of these two genes also cause their tail defects. Previously, studies have indicated that those traits usually involve through gene duplication or changes in the spatial temporal patterns of gene expression. Now that we wonder if the wind genes in seahorse account for its spatial tail development, so we conducted the following study in non-seahorse. By comprehensively analyze the genomic and transcriptomic data, we identified 22 wind genes numbers in seahorse. Interestingly, neither wind gene non-change nor spatial gene oxygen construction change were observed in the 22 wind genes. The motif distribution of the 22 wind genes also showed a conserved gene domain architecture for the wind genes. The phylogenetic analysis of the 22 wind genes show that the wind genes can be classified into 12 subfamilies, including the wind 1 to wind 11 and wind 16. Although several studies have shown that although several wind genes were duplicated, those seahorse spatial wind genes were observed when compared with other vertebrates. Besides, the sentinel analysis also showed that the wind genes show a conserved genomic organization in seahorse. Thus, we can get a brief summary here. The wind gene family in seahorse is conserved during seahorse evolution. The spatial tail morphogenesis will not be caused by wind gene duplication. So at the next steps, we detected the expression file of the wind genes during seahorse tail development process. Our results showed that 4 wind genes, wind 5A, 8A, 9B, and 11, were obviously fluctuated during seahorse tail development process. 3 different expression patterns were observed during this process. The wind 5A, wind 11 were highly expressed at a later stage. Interesting, wind 8A and wind 9B showed a completely opposite expression pattern during this process. Wind 8A prominently expressed at the early stage and kept decrease at the following stage. However, wind 9B kept increasing during the whole process. Thus, we could hypothesize that 4 wind genes may be the key candidate winds involved in seahorse tail development process. To confirm the hypothesis above, we further detected the expression of the 4 candidate wind genes by shoot hybridization. Our data show that wind 5A and wind 8A were accumulated at the seahorse tail bud during the former 3 stages, indicate the 2 wound genes may function as the seahorse tail bud formation. Interesting, compared with zebrafish, whose wound 8A gene mainly expressed at the early embryogenesis and degrades rapidly at the smith-genesis. Our seahorse wound 8A genes expressed for a long time at the later stage. For wound 11, the expression of wound 11 mainly a community at the anatomy indicate its potential role in the seahorse tail smith-genesis. As the wound 9B mainly expressed at the primary digest tract should indicate it was not associated with seahorse special tail development. Thus here we identified 3 wound genes, the wound 5A, wound 8A, and 11, were the key wound genes that involved in seahorse special tail development. To explore the potential function of the 3 key wound genes in seahorse tail development, the downstream 2 markings of wound signal pathway were evaluated after wound gene overexpression in seahorse CO9 invention. In this study, the seahorse exit mechanism mark gene SHH were upregulated in seahorse CO9 after overexpressing the wound 5A and wound 8A. Besides, the tail smith mechanism mark gene MYOD also upregulated in seahorse CO9 after overexpression of the wound 5A and wound 11. Besides, the 2 mark genes also showed a correlate expression of the wound genes during the seahorse embryogenesis. So the results support our hypothesis above, indicate that the wound 5A and wound 8A promote seahorse tail bud formation, and the wound 11 promote seahorse tail smith-genesis. Given the expression of the 3 key wound genes, partial overlap expressed during seahorse tail development, the potential interaction relationship of the 3 wound genes were detected. Our results show that a significant interaction relationship among the 3 wounds. In cells, overexpression of wound 5A, the expression of wound 8A are downregulated, and the expression of wound 11 were upregulated. In cells, overexpression of the wound 8A, just as the wound 11 was upregulated. It has no influence on the expression of wound 5A. In cells, overexpression of the wound 11, neither of the wound 5A nor wound 8A were influenced. Thus, we could draw such a deduced regulation law within the 3 wounds. There are similar results also reported in previously study. Finally, let's make a summary for this study. A comprehensive analysis of the vital tail development genes, the wound gene family, we found that the wound gene family is evolutionarily conserved in seahorse tail. In seahorse, the special seahorse tail would be not caused by the special gene duplication in seahorse. Also, we identified 3 Wind genes that are involved in seahorse special tail development. However, compared with zebrafish, just Wind 5A and Wind 11 were involved in the Smith jealousies in our seahorse, and a special expression change of the Wind 8A involved in this process, and an extended expression pattern of the Wind A may function to promote the tailbud formation, as well as by promoting the Wind 11 to promote tail summit formation. That would be contribute to the elongation of the special tail formation in seahorse. At last, I acknowledge my supervisor, Professor Lin Qiang. I also appreciate the help from Zhang Chunli professor, and also thanks for the group member in our lab. In addition, I thanks for the foundation project of National Natural Science Foundation of China and the Chinese Postdoctoral Science of Foundation. That's all. Thank you for your attention. Please ask me if you have any questions.
Thank you very much for that great talk. Does anyone have any questions for Bo?
I have a question for you, Bo. Do you think the extended period of Wind 8A expression could also be in other synaphids besides the seahorse, or do you think it only happens in seahorses?
Thank you. A good question. Before we started this experiment, we found the expression of wind genes are very important for the vertebrate body plan. As introduced by our in the background, the previously study have shown that the novel traits can be arised by the gene duplication or the special gene expression changes. So we observed these key genes in seahorse. We found that the Wind 8A gene is obviously extended expression pattern in seahorse embryo jealousies. So we conducted the experiment. Thank you.
Are there any other questions? If nobody else has a question, I'll ask a question. I was wondering, because obviously seahorses don't just have elongated tails, they also have the prehensile tail. Is this Wnt signaling controlling only the skeletal development, or do you have any idea whether it's also controlling the musculature that's required for prehensile tails?
Thank you. Of course, we don't exclude other genes involved in seahorse special tail developments. We just explain the special expression of wind genes. That may be one of the reasons for the elongation of seahorse. In seahorse, the Wind 8A mainly focus on its tail bud progenitus or stem cell formation, and the Wind 8A may promote the Wind gene of the expression of Wind 11. Thus, function at the summit jealousies. So they combine the function. Both of the wind genes combine and contribute to the special elongation of the seahorse tail. Thank you.
Excellent. Thank you very much.